All right, when we talk about the genetic code, the first thing that had to happen to understand how transcription and translation occurred is we had to crack the genetic code. So, when looking at DNA and RNA, we understand that there are nitrogen bases, ATGC if you're talking about DNA, or AUGC if you're talking about RNA. So the very first thing that happened when talking about the genetic code is we had to figure out how to read our DNA. So when we look at DNA, we had to crack this code. So we had to decipher it. So the first thing that we had to do is we had to determine how the 4 nucleotides which are going to be A, T, G, and C in DNA, could encode for 20 different amino acids. We had to figure out how DNA was used to eventually make protein. Amino acids are the building blocks for protein. So the very first thing is we have 2 scientists. The first 2 scientists to actually start looking at DNA and how it could encode for amino acids was Francis Crick and Sidney Brenner. So these guys determined that DNA is read in sets of 3. In sets of 3 nucleotides for each amino acid. So, the very first thing that we had to do when talking about DNA, of course DNA is going to be unzipped. We had to understand that DNA is read in sets of 3. You can make these up. GGG, CCC, AAA, TTT. I'm just using the same letters. They're read in sets of 3. You will always see me write these in sets of 3. GCA, TAC, GAT, TAC. This is called a codon. When you talk about, I'm going to put a star by this, a codon is a set of 3 nucleotides that is specific to a particular amino acid. So when we talk about codons, codons can be either DNA or RNA as long as they're written in 3s. Now we know that during transcription we have to have mRNA to basically figure out your amino acids. So even when we use DNA, we have to understand that you have to transcript your DNA into RNA. So during transcription, when you produce your mRNA, these are going to be complementary to DNA. These are also read in codons. These are complementary to the DNA. So CCC, GGG, U, because there's no T's in RNA, and A. These are also called codons. So the next scientist had to figure out basically the RNA sequences and which amino acids that they code it for. So this is getting to the genetic code. A scientist by the name of Marshall Nierenberg identified RNA codons. I should put mRNA codons that specify amino acids. Now there's different variations of the genetic code, which is the mRNA, to help you figure out which amino acids is used. Some people use a box, others are going to use a circle, but basically he figured out all the mRNA codes, all the mRNA codes that can code for amino acids. So, what he determined: mRNA molecules of specific 3 base sequences are used to determine the amino acid sequence of a protein. That is key. mRNA molecules are used to determine amino acid sequences when we use them in 3s. CCC is going to encode for an amino acid. GGG, UUU, AAA. mRNA is used to determine your amino acid sequence. Now, something else that he discovered is that there were 64 possible mRNA codons. Something he also discovered is that there are stop codons. Stop codons are going to stop translation or stop the production of translation. There's 3 of them that are stop codons: UAA, UGA, and UAG. Stop codons in the genetic code are used to stop or terminate translation. So anytime you get UAA, UGA, UAG, you write stop. Now, start codon. There's only one. So it's the code. This is the genetic code. We're cracking the code. There's one codon that is a start codon, and its sequence is AUG. This is used to start translation, which is to make a protein. Now, when we talk about AUG, methionine is the start amino acid, which we write as MET, used to start translation. So literally, it's going to be the first amino acid that is always built every time. It's the start amino acid. AUG is used to figure out which amino acid. So you use that little chart, and basically it's always going to be mRNA. mRNA is always going to help you figure out which amino acid you're going to use. So you would use your genetic code chart to figure out which amino acid would be translated for each of these codons. So when we talk about the genetic code, in conclusion, the genetic code is the rules that dictates correspondence between mRNA molecules and amino acid sequences. You always use mRNA.